Hi, this is Dr. Muscatello again. In Chapter 3, we are going to cover how the immune system develops and how the cells are selected so that they can participate in an active immune response.
So I'd like to thank Dr. Muscatello for doing an excellent job for setting me up to discuss with you the development and selection of our B and T lymphocytes. I really want to reinforce that gene rearrangement of the receptors drives development of the lymphocytes. And development always begins within the bone marrow. And the goal, or I should say one of the goals of the gene rearrangement is to ensure that each receptor has a unique idiotype or specificity. Recall that any one cell always has one idiotype, meaning it has a single specificity. Because development begins within the bone marrow, it is considered to be one of the primary lymphoid organs. We will talk about the fact that B cells will remain within the bone marrow to complete a majority of their development, while T cells, which are those high maintenance bad boys, have to exit the bone marrow and travel to the thymus to undergo their gene rearrangement, again, which will drive its development. After the B cells have completed development and prior to exiting the bone marrow, one other thing must come into consideration. Recall that the gene rearrangement is a random rearrangement of those various fragments, so you can establish a variety of different idiotypes. Well, because of the randomness of those rearrangements, you have to assume that there are going to be numerous receptors that are going to recognize self. And when I am recognizing self, I have to be able to determine which cells are self-reactive and, to the best of the ability, destroy as many of those cells before they escape from the bone marrow. So everybody needs to take a deep breath for me right now. This is probably one of the busier figures within the text itself, and it's probably one of the more important. It has a lot of information that gets heavily tested on the step one. And a lot of the information you've already been introduced to by Dr. Muscatello. So what you're going to need to be aware of are the different steps in B cell development. All cells are going to be derived from that multipotent stem cell. And if you recall back in Chapter 2, I defined what a multipotent stem cell was, and we discussed one of the markers found on that multipotent stem cell, which will be CD34. As a B cell begins to rearrange its heavy chain, remember the rearrangements? I go D to J and then V to D to J. That is going to change the stage of development from the lymphoid stem cell to the progenitor B cell arena. Once heavy chain gene rearrangement has been completed, you start light chain gene rearrangement, and that is going to bring us to our pre-B cell stage. So we've gone from being a lymphoid stem cell to a progenitor B cell, and then a pre-B cell. And again, that advancement through those stages are driven by the gene rearrangement. Something very important that needs to be stated about our pre-B cells is that they have a unique marker. They have very high levels of cytoplasmic mu heavy chain, and that is going to be able to essentially stage B cell development. So if you have developed something like a B cell leukemia or lymphoma and they give you back a report that says there were high levels of cytoplasmic mu heavy chain, then you're going to be able to very quickly stage it at the pre-B cell level. Once you have completed heavy chain gene rearrangement and light chain gene rearrangement, the cell will express the first isotype on its surface, the IgM. And now we're going to refer to that B cell as being immature. And it is the immature B cell that is going to exit the bone marrow and enter into the periphery. And it is that transition from the bone marrow into the periphery that is going to cause the cell to, again, advance to the next stage, which is going to be the mature B cell stage. At the mature B cell stage, you are going to see co-expression of the IgM and IgD on the surface, again, defining a mature or sometimes called naive B cell. And I would also remind you that that co-expression is due to the fact that you have alternative splicing of the mu and delta constant regions. Now, that pretty much is the end of development. The rest of the stages are going to be dependent upon a cell engaging their antigen, which is going to take place in the secondary lymphoid tissues. So once a B cell has bound to its antigen through its B cell receptor, which is going to be composed of the IgM, associated signal transduction complexes also, you are going to see that the B cell is then going to differentiate into one of two cells. A majority of the differentiation is going to be through plasma cells, and those plasma cells are going to be, again, the cells that secrete our antibodies. A much smaller group of B cells are going to become memory cells. And we're going to talk a lot about memory cells when we talk about vaccination. I think it's of important note, though, to state that memory B cells are going to look a lot like our mature naive B cells, but rather than expressing IgM and IgD on their surface, they will only express IgG, IgA, or IgE, indicating that they have undergone isotype switching due to activation. Now, the last part of this slide that I want you to recognize is the expression of the various enzymes associated with gene rearrangement, as well as some of the surface markers. So this should be sort of intuitive. If I am undergoing gene rearrangement, one would expect that I would have RAG expression, and that the expression of my RAG1 and RAG2 would continue until light chain gene rearrangement is complete. We also have the expression of that TDT. Remember, the TDT involved in junctional diversity. Again, once I begin rearrangement of my gene fragments for the heavy chain, I will already be expressing TDT. And I also want to reiterate that the activity of TDT pretty much diminishes after I have my heavy chain gene rearrangement. A couple of other markers that are going to be important for my B cells is the fact that B cells are one of three very important professional antigen presenting cells that express the MHC class II on their surface. And we're going to go a lot more into our MHCs in just a little bit. We also have what I consider three of the most important markers after the immunoglobulins on our B cell surface are CD19, CD21, remember those were part of the components of the B cell receptor, as well as CD20. So when anybody ever asks you, what are B cell markers? It's easy, CD19, 20, 21, you just had to count. There's also going to be on the surface a marker called CD40. I do want to express to you that CD40 is not unique for B cells, but it's going to be extremely important when we have T cell activation. So one should really ask themselves, how is it that I am able to determine which B cells are self-reactive? And that is going to be one of the roles of the bone marrow stroma and the interdigitating cells. Basically, any strong interaction between a B cell and the stroma or the interdigitating cells is going to cause the cell to either undergo apoptosis, or they can potentially undergo what is known as anergy. So essentially you need to think about elimination as being, again, through deletion via apoptosis or through inactivation by anergy. Please recognize that the anergic B cells will actually exit the bone marrow. So how is it they are non-functional? Well, anergic B cells will downregulate their expression of IgM on the surface and express very, very high levels of the IgD isotope. Recall that IgD is simply a developmental isotype and has no effector function, so it essentially renders the cell anergic. The process of selecting out self-reactive B cells within the bone marrow is to minimize the number of self-reactive B lymphocytes that are actually going to make it out into the periphery. Please make no mistake, though, that there is still a significant number of self-reactive B cells that will enter into circulation, so there obviously has to be another mechanism of inactivation. When a cell is non-self-reactive, in other words, it is not reactive to self-antigen, it would be referred to as being tolerant, meaning I tolerate myself or do not react to myself. Another term that is used is that they are selectively unresponsive. So now let's get on to talking about those high-maintenance T lymphocytes and their development. Again, T lymphocytes, or I shouldn't even say T lymphocytes, let's go ahead and just say progenitor cells are going to exit the bone marrow, and they are going to get into the bloodstream and go to the thymus. And it is within the thymus that my T cells are going to undergo development. As with our B lymphocytes, development of T cells are driven by the gene rearrangement of the receptor components. So the thymus, just like the bone marrow, is going to be considered a primary lymphoid organ. Now, before a cell enters into the thymus, it is going to be referred to by a couple of different names. It is either referred to as a prethymic cell, makes lots of sense, doesn't it? Or you can refer to them as double negative T lymphocytes. So what does the double negative stand for? The double negative simply refers to cells that are going to enter into the thymus but do not express the cell surface markers CD4 or CD8 on their surface. Well, welcome to the thymus. Remember, with our progenitor cells, they are going to be considered prethymic or double negative before entering into the thymus. Once they get into the thymus, they are going to enter into the subcapsular area of the cortex. It is here that we find the most immature T cells, which now have a new name, because why not? They're very high maintenance. So within the thymus, we now are going to refer to our T cells as being thymocytes. They will begin their gene rearrangement within the cortex, starting first with the beta chain, followed by the alpha chain. As they continue to develop, they will move from the cortex into the medullary region. And as they make that movement, they are going to undergo their selection process, which is unfortunately a little more complicated than B cell selection, but hoping it's going to make a lot of sense when we go through it. As the T lymphocytes undergo gene rearrangement of their beta and alpha chains within the cortex, they're going to also start co-expressing cell surface markers like the CD3 complex. And does anybody remember what the CD3 complex does? And yes, you are right, signal transduction. You're also going to start co-expressing the CD4 and the CD8. We can now call these cells double positive versus the double negative cells, which were prethymic. The T cell receptor is a very unique receptor in that it is designed to recognize two different entities. The first thing that it needs to recognize is going to be a peptide. But it can't just recognize a peptide. That peptide actually has to be coupled to and presented by a unique group of molecules referred to as the major histocompatibility complex antigens, better known as MHCs. Now, the MHCs are typically found on the surface of antigen-presenting cells, and as we're about to discuss, our MHCs come in two flavors. They come in a class 1 and they come in a class 2.
T cell development is a very stringent process that occurs within the thymus. And as these T cells are developing within the thymus, they are trying to learn the difference between self versus non-self, and hopefully eliminating any T cells that would be potentially self-reactive and lead to autoimmune disease. There is actually a two-step selection process for these T cells. One is called positive selection, and the other is called negative selection. But the gist of the selection process is that because T cells always have to see antigen in the context of an MHC molecule, within the thymus during development and selection, the T cells have to be able to recognize all of the different MHC types, which will actually guide their differentiation into T helper cells or cytotoxic T cells. And they also have to sort of know when to stay away and not become activated, because those cells might potentially be autoreactive.
The MHC complex itself is a group or collection of highly polymorphic genes that are located, clustered together on the chromosome 6. Recall back when you were talking about your genetics, what the word polymorphic meant. It really is just a representation of multiple alleles within a population. So depending on what population you're looking at, you're going to have different representation of your MHCs. And please recall that we do have two major classes. These aren't the only classes, but these are the two that you will know for this step 1. We have class 1 and class 2. Now, both class 1 and class 2 are both expressed at a very, very, very high density on the stromal cells of the thymus. Now, we're going to find that that is not the case on the cells with the remaining tissue in the body. There's actually a very unique distribution of class 2 molecules versus class 1. Here's an area of immunology that I find fairly interesting. A lot of the nomenclature of immunology is already difficult, so let's go ahead and call two things by two different names that do the exact same thing. Really quick story is that there were two competing research groups that were studying the same proteins, but one was in a mouse model and one was in a human model. So when the mouse model group published, they referred to them as the MHCs. When the human group published, they called them HLAs or human leukocyte antigens. As clinicians, you use these two terms interchangeably. So that puts you at a slight disadvantage because there are some nuances about HLAs that you need to be aware of. Now remember, I divided my MHCs up into class 1 and class 2. You do the same with your HLAs, but there are multiple genes encoding for the class 1s in humans. So with my class 1s, I have represented HLA-A, HLA-B, and HLA-C. Those are the highest level of polymorphic class 1 HLAs. I also have class 2, and class 2 are going to be primarily the HLA, DP, DQ, and DR. Now I want you to immediately say, why did she skip DM? Well, I want to point out to you that the class 2 genes that will be able to bind and present peptide are only the DP, the DQ, and the DR. The HLA-DM does not bind to peptide and present it on the surface. And as Dr. Moscatello is going to go through later, what you're going to see is that HLA-DM is actually going to be very important for chaperoning the loading of peptides onto the other class 2 molecules. Now, you might be asking yourself, well, how am I going to remember the differences between the class 1 HLAs and the class 2? So I always go back to a song that I heard in my childhood. And I love the Jackson 5. So if you can sing it in your head, here's a great song. We've got A, B, C, easy as one. And then I just want you to stop counting. So your HLAs A, B, and C are your class 1s. The nice part is that your class 2s all start with D. Let's go ahead and look a little more closely at our class 1 and class 2 MHC molecules. And again, reminding yourself of the HLAs that belong to these different classes. Class 1 MHCs are found on any cell in the body that has a nucleus, which means the only cell that you should not find any expression of class 1 would be on your red blood cells or erythrocytes. Another important thing I want you to note is that those class 1 molecules, your HLA A, B, C, have 2 alleles. And each allele of those genes is going to be codominantly expressed. This does not fall under normal Mendelian inheritance patterns. So what does the MHC class 1 molecule look like? Well, it is actually a single transmembrane alpha chain, and that alpha chain is going to be associated with a beta-2 microglobulin, which is going to actually serve the role of helping maintain the structure of the MHC class 1 molecule, as well as helping maintain the binding groove where you're going to place the peptide. The actual binding groove is found completely within the alpha chain, and it's a relatively small groove, as you can see over here in this 3D representation. So the average size of the peptides that are able to be loaded onto an MHC class 1 typically range from about 8 to 10 amino acids in length. Our class 2 molecules are only found in a limited number of cells within the body, and although there are several different types of cells that will express these molecules, there are only 3 that we really need you to know for the step 1. Those are going to be our macrophage, our dendritic cells, and our B lymphocytes. Keep in mind that these 3 cells will not only express on their surface our class 2 MHC molecules, but also class 1. And why is that? It's because they are also nucleated. As we saw with the MHC class 1s, our class 2 molecules are also expressed codominantly. The actual structure of the MHC class 2 molecule is 2 separate transmembrane proteins, one alpha and one beta, with both chains contributing to the structure of the peptide binding groove. Now, the peptide binding groove of the class 2 molecules tends to be much more accommodating than that of the class 1. And there's a lot of variability in the peptide size that can go onto your class 2 molecules. And I like to think about it like being a hot dog bun. You can either have a really tiny hot dog on that bun, or you can have a foot long that spills out the side. So again, variety of different peptides. But in general, they typically are a couple of dozen amino acids in length. After completion of T cell development, we have to then go on to select out any of our self-reactive T lymphocytes. Unlike B lymphocyte selection, this is actually a more complex approach and actually involves a 2-step selection process. Now, when we look at our stroma of the thymus, recall that they express very, very high levels of MHC class 1 and class 2, and those MHC molecules are all loaded with self-peptide. And it is at this stage that we have to determine if the T cell receptor is capable of recognizing 2 things. 1, self-MHC. The next is determine whether or not the T cell receptor can bind to self-peptide. So the first stage of selection is referred to as positive selection. And with positive selection, what you're really measuring is if the T cell receptor is capable of binding to the MHC. If a T cell receptor interacts with an MHC with low affinity, they are going to survive positive selection and go on to negative selection. Now, if I have a T cell that expresses a T cell receptor that does not recognize MHC, it is going to undergo apoptosis, and this is referred to as a failure of positive selection. So only those cells that survive positive selection will go on to the next stage. These cells are then going to be subjected to negative selection. And in negative selection, you are trying to determine if that T cell receptor recognizes self-antigen, which in this case is not a good thing. So if a cell has a T cell receptor on it that binds very strongly to both the MHC and to the peptide being presented, it is going to undergo apoptosis. So these cells will die during negative selection. So you have a twofold end result. The first is that you have a T cell receptor that can bind to MHC, albeit very low affinity to the MHC. And two, you have a T cell receptor that does not bind to self-peptide. At the completion of T lymphocyte selection, a majority of the cells that enter the thymus will die within the thymus. Those that have survived have undergone positive selection as well as negative selection, and are then going to be termed single positive. Single positive indicates that the cell either expresses CD4 on their surface or they express CD8 on their surface. Please recall that CD4 is a co-receptor molecule on a T cell that recognizes and binds to MHC class II. CD8, on the other hand, is a co-receptor found on a T cell that is going to interact and bind to class I. CD4 cells in general will become what are known as helper T cells, where our CD8 positive cells will eventually differentiate into cytotoxic T lymphocytes. There's a very interesting subset of T cells that arrives during the selection process. While most of your self-reactive T cells are going to undergo apoptosis within the thymus, there is a very small population of CD4 positive T cells that want so desperately to survive that they are willing to change their focus. We refer to these cells as regulatory T cells or Tregs. The Tregs are going to exit the thymus and serve in maintaining peripheral tolerance. You're able to identify Tregs based on the expression of CD4 on their surface, as well as a unique marker CD25. Additionally, Tregs have a unique transcription factor called FOXP3. Upon activation of a Treg, they are going to secrete very high levels of the cytokines IL-10 and TGF-beta. Both of these are inhibitory to Th1 cells, which drive inflammation, so they essentially shut off an inflammatory response. There's also a strong correlation in the number of functioning Tregs and autoimmunity. So if I have very low levels of Tregs, you are going to find an individual is much more prone to developing autoimmune disease than those who have normal levels of Tregs. This particular figure outlines the different stages of T cell development, as well as looking at the markers associated with the various stages. Recall that we have our cells that have exited the bone marrow and are traveling to the thymus. These again referred to as prethymic, or remember, we can also call them double negative, as they do not express on their surface CD4 or CD8. If you're looking for a marker of your prethymic or double negative cells, the best marker that you're going to have is going to be that TDT. And TDT is a hot marker in the step 1 because it is typically associated with ALL. So please take note that even though TDT was discussed here primarily associated with junctional diversity, you might see it appear in a pathology question. Now, as the T cells, or prethymic cells, enter into the cortex of the thymus, they are going to start undergoing gene rearrangement. And in order to undergo gene rearrangement, you're going to see the increased expression of RAG1 and RAG2. Also, as I start to rearrange my genes, I'm going to start to see the expression of some additional markers like CD2. Now, I don't want you to get too concerned about CD2. CD2 is an oldie but goldie T cell marker, and it was an adhesion molecule, or still is an adhesion molecule, but for some reason, this is a particular marker that shows up in Q banks. Not on the USMLE, but on Q banks. So we wanted to mention it here so you didn't have that moment of panic that you forgot something. As soon as I start rearranging my genes, I'm also going to start expressing CD3 on the surface, my signal transduction complex. Also, I will express my CD4 and CD8. So now my double negative cells have become double positive cells. And a little bit later on, you will see the appearance of the T cell receptor. As development continues, the cells are going to start moving from the cortex into the medulla and undergo that positive and negative selection. After the completion of positive and negative selection, your T cell had to make a decision. Did it recognize MHC class II and therefore preferentially express CD4? Or did it recognize MHC class I and preferentially express CD8? So cells that have undergone selection can appropriately be termed single positive. Again, they're either single positive for CD4 or they are single positive for CD8. And at this point, the cells can exit into the periphery.